Hello friends, I am Dr. Varsha Kashav, Professor Swami Vivekanand Institute of Pharmaceutical Sciences, SVN University, Sagar. Today in this session we will learn antifungal agent part 1 and 2. This script is divided into 5 different modules. Module 1 is introduction, module 2 antifungal drugs like antibiotic Griseofulvin, module 3 is antifungal drugs like Candicidine and nystatin. Module 4 is synthesis of antifungal agent like salicylic acid, miconazole, etc. Then module 5 is summary and conclusion. Now let us start with module 1 is introduction. An antifungal medication is a pharmaceutical fungicide used to treat and prevent mycosis such as athlete's foot, ringworm, candidiasis, serious systemic infection such as cryptococcal meningitis and other. Many topical antifungal have been available since the antiseptic era. Two important antibiotic Amphotericin B deal with systemic mycosis and Griseofulvin. Supplement attack on dermatophytes were introduced around 1960s. Fungal microorganisms are believed to damage the cell membrane, leading to a loss of essential cellular component. This may result in inflammation of the infected tissues, which in some cases may be several. Antifungal agent counter mycosis by both fungistatic and fungicidal action. Fungistatic action occur when a drug prevents the fungi reproducing with the result that it dies out naturally, whilst fungicidal action kill the fungi. The suffix static and side are widely used to indicate these general type of action. Fungal microorganisms differ from other microorganisms in that they consist of eukaryotic cell with a chitin cell wall. This means that their chemical structure and biochemistry are similar to those of humans. Consequently, it is more difficult to design drug that would selectively target these fungi. However, these are some differences that can be utilized. For example, human cell membrane contain cholesterol, but those of fungi contain ergosterol. A number of antifungal drugs are believed to act by blocking the biosynthesis of ergosterol in fungi. Antifungals work by exploiting differences between mammalian and fungal cell to kill the fungal organism with fewer adverse effect to the host. Unlike bacteria, both fungi and human are eukaryotes. Thus, fungal and human cell are similar at the biological level. This makes it more difficult to discover drugs that target fungi without affecting human cells. As a consequence, many antifungal drugs causes side effects. Some of these side effects can be life-threatening if the drugs are not used properly, apart from side effects like liver damage or affecting estrogen level. Many antifungal medicines can cause allergic reactions in people. For example, the azole group of the drug is known to have caused anaphylaxis. There are also many drug interactions. Patient must read in detail the enclosed data sheet of the medicine. For example, the azole antifungal such as ketoconazole or itraconazole can be both substrates and inhibit of P-glycoprotein, which excrete toxin and drug into the intestine. Azole antifungal also are both substrates and inhibitors of the cytochrome P450 family cytochrome P3A4, causing increased concentration when administering, for example, calcium channel blockers, immunosuppressant, chemotherapeutic drugs, benzodiazepine, tricyclic antidepressant, macrolides and SSRIs. Before oral antifungal therapies are used to treat nail disease, a confirmation of the fungal infection should be made. Approximately half of the suspected case of fungal infection in nails have a non-fungal cause. The side effects of oral treatment are significant and people without an infection should not take these drugs. Now classification. The first one class is antibiotic. For example, paulines, Amphotericin B, nystatin, hemicin, natamycin. Second class is heterocyclic benzofuran and Griseofulvin. Next class is antimetabolites, for example, fucytosine. Next class is azoles. Their subclasses is like first is imidazole, for example, clotrimazole, iconazole, miconazole, oxyconazole, ketoconazole, etc. Next one class is triazole, for example, fluconazole, itraconazole, voriconazole. The next one main class is allylamine, for example, terbinafine. Next one class is other topical agent, tolnaftate, undecyclinic acid, benzoic acid, quinidochlor, cisclopirox, olamine, butanafine, sodium thiosulfate. Next one is benzoic acid, has antifungal properties but must be combined with a keratolytic agent such as in Whitefield ointment. The next in ciclopirox, it is a hydroxypyridone antifungal that interfere with active membrane transport, cell membrane integrity and fungal respiratory process. It is most useful against tiny versicolor. Next one is flucytosine or 5-fluorocytosine, an antimetabolites pyrimidine analog. Next one is Griseofulvin, bind to polymerized microtubule and inhibit fungal mitosis. Next one is haloprogen, discontinued due to the emergence of more modern antifungal with fewer side effects. Next one is tolnaftate, a thiocarbamate antifungal which inhibit fungal squalene epoxidase. Mechanism of allylamine like terbinafine. Next one is undecyclinic acid, an unsaturated fatty acid derived from natural castor oil, fungistatic, antibacterial, antiviral and inhibit candida morphogenesis. Next one is crystal violet, a tertiary methane dye. It has antibacterial, antifungal and anthelmintic properties and was formerly important as a topical antiseptic. Then balsam of Peru has antifungal properties. Now module 2, antifungal drugs like antibiotics, Griseofulvin. Pauline antibiotics. The name Pauline is derived from their high double bonded structure. Amphotericin B is described as a prototype. Amphotericin B, it is obtained from Streptomycinodosis. The Paulines possess a microcyclic ring, one side of which has several conjugated double bond and is highly lipophilic, while the other side is hydrophilic with many OH groups. A polar amino sugar and a carboxylic acid group are present at one end in the same. They are all insoluble in water and unstable in aqueous medium. The Paulines have high affinity for ergosterol present in fungal cell membrane. Combined with it, get inserted into the membrane and several Pauline molecules together orient themselves in such a way as to form microspore. The hydrophilic side form the interior of the pore through which ions, amino acid and other water soluble substances move out. The micropore is stabilized by membrane sterol which fill up the space between the A and B molecule on the lipophilic side chain on the outer surface of the pores. Thus, cell permeability is markedly increased. Cholesterol present in host cell membrane closely resemble ergosterol. The Pauline bind to it as well, through with lesser affinity. Thus, the selectivity of action of Pauline is low, and Amphotericin is one of the most toxic systematically used antibiotic, through it is the less toxic Paulines. Bacteria do not have sterol and are unaffected by Paulines. It has been found that amphotericin enhance immunity in animal, and this may aid immunocompromised individual in handling fungal infection. Dermatophytes are inhibit in vitro, but concentration of amphotericin attained in infected skin are low and ineffective. It is fungicidal at high and static at low concentration. Resistance to amphotericin during therapy has been rarely noted among Candida in a selected group of leukopenic cancer patient, but it is not a problem in the clinical use of the drug. Amphotericin B is also active on various species of Leishmania. Their uses: amphotericin B can be applied topically for oral, vaginal and cutaneous indidiasis and otomycosis. These are the chemical name of amphotericin B and their structure. Biosynthesis of amphotericin B. The natural route to synthesis include polyketide synthase component. The carbon chain of amphotericin B are assembled from 16C2 acetate and 3C3 propionate unit by polyketide synthase. Polyketide biosynthesis begin with the decarboxylative condensation of a dicarboxylic acid extender unit with a starter acyl unit to form a beta ketoacyl intermediate. The growing chain is constructed by a series of Claisen reactions within each module. The extender unit are loaded onto the current ACP domain by acetyltransferase. The ACP bound elongation group react in a Claisen condensation with the Ks bound polyketide chain. Ketoreductase, dehydratase and enoyl reductase enzyme may also be present to form alcohol. Double bond or single bond after cyclization. The macrolactone core undergoes further modification by hydroxylation, methylation and glycosylation. The order of these processes is unknown. Second, Griseofulvin. Griseofulvin is an orally administered antifungal drug. It is used both in animal and in human to treat fungal infection of the skin, commonly known as ringworm and nail fungus. It was discovered in the culture broth of certain species of the mold Penicillium in 1939. It is on the World Health Organization list of essential medicines, the most important medications needed in a basic health system. It is produced industrial by fermenting the fungus Penicillium patulum. Griseofulvin is used orally only for dermatophytosis. It is effective topically. It is reserved for cases with nail, hair or large body surface involvement. The drug binds to tubulin, interfering with microtubule functions, thus inhibiting mitosis. It binds to keratin in keratin precursor cell and make them resistant to fungal infection. The drug reaches its site of action only when hair or skin is replaced by the keratin Griseofulvin complex. Griseofulvin then enter the dermatophytes through energy dependent transport processes and bind to fungal microtubules. This alter the process for mitosis and also underlying information for deposition of fungal cell wall. This is the chemical name of Griseofulvin: 2,6,6R 7-chloro 2,4,6 trimethoxy 6-methyl 3H4H spiro 1-benzofuran 2,1-cyclohextuene 3,4-dione. And this is the structure. Now, biosynthesis of Griseofulvin. The first step in the biosynthesis of Griseofulvin by P-Griseofulvin is the synthesis of the 14-carbon poly-beta keto chain by a type 1 iterative polyketide synthase via iterative additions of 6-malonyl coenzyme A to an acyl coenzyme A starter unit. The 14-carbon poly-beta keto chain undergoes cyclization, aromatization using cyclase, aromatase respectively through a Claisen and aldol condensation to form the benzophenone intermediate. The benzophenone intermediate is then methylated via S-adenosyl methionine twice to yield Griseofulvin C. The Griseofulvin C is then halogenated at the activated site ortho to the phenol group on the left aromatic ring to form Griseofulvin B. The halogenated species then undergoes a single phenolic oxidation in both rings, forming the 2 oxygen diradical species. The right oxygen radical shift alpha to carbonyl via resonance, allowing for a stereospecific radical coupling by the oxygen radical on the left ring, forming a tetrahydrofuranone species. The newly formed Grisene skeleton with a spiro center is then orthomethylated by SEM to generate dihydrogriseofulvin. Ultimately, a stereoselective reduction of the olefin on dihydrogriseofulvin by NADPH afford Griseofulvin. Now, module 3, antifungal drug, Candicidine, Nystatin. Now, first is Candicidine. Candicidine is an antifungal compound obtained from Streptomyces griseus. It is active against some fungi including Candida albicans. Candicidine is administered intravaginally in the treatment of vulvovaginal candidiasis. This bioactive compound was named Candicidine because of its high activity on Candida albicans. This is the chemical name of that drug, 33 3-amino 3,6-dideoxy D-menopyranosyl oxo 17 6,4-aminophenyl 4-hydroxy 1,3-dimethyl 1,6-oxohexyl 1,3,5,7 37 pentahydroxy 18-methyl 9,13,15 trioxo 16,39 dioxa bicyclo 33,3,1 nanotriconta 19, 21, 23, 25, 27, 29, 31 haptene 36 carboxylic acid. Microorganism and growth condition Streptomyces griseus, the strain producing Candicidine, growth in a medium supporting high antibiotic production which contained soya peptone 25 gram, glucose 65 gram and zinc sulfate 0.5 mm. The culture were incubated at 32 degrees centigrade in triple baffleted 250 ml flask containing 50 ml of medium in a Gallenkamp orbital shaking incubator operating at 225 rpm. The production flask were inoculated with 5 ml of 24-hour inoculum growth in a medium containing yeast extract 10 gram and glucose 10 gram. The second is Nystatin. Nystatin solid under the brand name Mycostatin among other. It is an antifungal medications. It is used to treat candida infection of the skin including diaper rash, thrush, esophageal candidiasis and vagina yeast infection. It may also be used to prevent candidiasis in those who are at high risk. Nystatin may be used by mouth in the vagina or applied to the skin. Common side effects when applied to the skin include burning, itching and a rash. Commonly side effects when taken by mouth including vomiting and diarrhea. During pregnancy, use in the vagina is safe, while other formulations have not been studied in this group. It works by disrupting the cell membrane of the fungal cell. Medical use: skin, vagina, mouth and esophageal candida infection usually respond well to treatment with Nystatin. It is available in many forms. Oral Nystatin is often used as preventive treatment in people who are at risk for fungal infections such as AIDS patient with a low CD4 plus count and patient receiving chemotherapy. It has been investigated for use in patient after liver transplantation, but fluconazole was found to be much more effective for preventing colonization invasive infection and death. It is also used in very low birth weight, below 1500 gram infant, to prevent invasive fungal infection. Although fluconazole is the preferred treatment, the rate of invasive fungal infections and also reduce death when used in these babies. Guidelines state that the use of these agents should be restricted to extremely low birth weight infant, below 1000 gram, in a neonatal intensive care unit with high baseline of fungal infection. Liposomal nystatin is not commercially available, but investigational use has shown greater in vitro activity than colloidal formulation of amphotericin B and demonstrated effectiveness against some amphotericin B resistant form of fungi. It offers an intriguing possibility for difficult to treat systematic infection such as invasive aspergillosis or infections that demonstrated resistance to amphotericin B. Additionally, liposomal nystatin appear to cause fewer cases of and less severe nephrotoxicity than observed with amphotericin B. This is the chemical structure of that drug: 1S, 3R, 4R, 7R, 9R, 11R, 15S, 1, 6R, 17R, 18S, 19E, 21E, 25E, 27E, 29E, 31E, 33R, 35S, 36R, 37S, 33, 3-amino 3,6-dideoxy, beta L menopyranosyl, oxyl 1, 3, 4, 7, 9, 11, 17, 37 octahydroxyl, 15, 16, 18 trimethyl, 13 oxo, 14, 39 dioxa bicyclo, 33, 3, 1 nona triaconta, 19, 21, 25, 27, 29, 31 hexane, 36 carboxylic acid. Now the biosynthesis of nystatin. Nystatin A1 is biosynthesized by a bacterial strain, Streptomyces neuracil. The structure of the active compound is characterized as a Pauline macrolide with a deoxy sugar, D-mycosamine, and aminoglycoside. The genomic sequence of nystatin reveals the presence of the polyketide loading module, 6 polyketide synthase module, nystatin B, nystatin C, nystatin I, nystatin J, and nystatin K, 2 thioesterase, nystatin K and nystatin E. Thus, it is evident that the biosynthesis of the macrolide functionality follows the polyketide synthase pathway. Following the biosynthesis of the macrolide, the compound undergoes post-synthetic modifications, which are added by the following enzymes: GDP mannose dehydratase, P450 monooxygenase, aminotransferase, and glycosyl transferase. The biosynthetic pathway is through to proceed as shown to yield nystatin. Now, module 4, synthesis of antifungal agent, salicylic acid, miconazole, etc. First one is salicylic acid. Salicylic acid is also a precursor to acetyl salicylic acid or aspirin. Acetyl salicylic acid, a ester derived derivative of salicylic acid, is much more commonly used than salicylic acid for pain relief because the parent compound can irritate the lining of the stomach. In this synthesis, methyl salicylate is the starting material or precursor and salicylic acid is the synthetic target. If the precursor goes well, we will be able to refer to salicylic acid as the product. The difference in the two structure is the single functional group in methyl salicylate. On the left, the benzene ring is substituted by two functional group, a hydroxy group and an ester group. In salicylic acid, the ester group has been replaced by a carboxylic group. Now, miconazole. Miconazole is imidazole type antifungal. When miconazole was first released, it was hoped it would replace amphotericin B as a parenteral antifungal agent. It was soon discovered, however, that miconazole not only failed to offer the same efficacy as amphotericin B but also possessed intrinsic toxicity after fourth administration. While miconazole is still marketed in a parenteral formulation, it is most commonly used topically and intravaginally. Miconazole was approved by FDA in 1974. It was one of the first azole antifungal agent to become widely used for treating mycosis. Miconazole is a topically used azole like other azole antifungals. Miconazole exerts its effect by altering the fungal cell membrane. Miconazole inhibits ergosterol synthesis by interacting with 14A demethylase, a cytochrome P450 enzyme that is necessary for the conversion of lanosterol to ergosterol, an essential component of the membrane. Inhibition of ergosterol synthesis results in increased cellular permeability, causing leakage of cellular content. Miconazole does not appear to have the same effect on human cholesterol synthesis. Miconazole is the derivative of miconazole. It is used of synthetic origin and belong to azole. It belongs to antifungal pharmacological group. The molecular weight of miconazole is 479.10. Miconazole is mainly used externally for the treatment of athlete foot, ringworm and jock itch. Internal application is used for oral or vaginal thirst. The oral gel may also be used for lip disorder, angular cheilitis. In the UK, miconazole may be used to treat neonatal oral thirst, while the alternative nystatin is only licensed for patients over the age of 1 month, but drug interaction are possible. Unlike nystatin, some miconazole is absorbed by the intestinal tract when used orally. This may lead to drug interaction. Interaction are possible with anticoagulant, phenytoin, terbinafine, some newer atypical antipsychotic, cyclosporin and some statin used to treat hypercholesterolemia. Now next drug is iconazole. Iconazole used as a nitrate salt. It is an antifungal medication of the imidazole class. It is used as cream under the brand name Spectazole, ecostatin, Peverly and Pebisone, consisting of the combination iconazole triamcinolone to treat skin infections such as athlete foot, tinea, pityriasis, versicolor, ringworm and jock itch. It is also sold in Canada under the brand name ecostatin as vaginal ovule to treat vaginal thirst. In Asia, it is sold under the brand name Index G, while in Thailand it is sold as Ecosone by Qualimed. Iconazole nitrate exhibit strong anti-feeding properties. Now the chemical name of the drug: 1,2,4-chlorophenyl methoxy 2,2,4-dichlorophenyl ethyl 1H imidazole. Imidazole devoid of the nitro group no longer have any antiprotozoal activity. However, such drugs are effective antifungal agent. Now the synthesis: alkylation of imidazole with bromoketone prepared from OP-dichloroacetophenone affords the displacement product. 3. Reduction of the ketone with sodium borohydride gives the corresponding alcohol. 4. Alkylation of the alkoxide from that alcohol with parachlorobenzyl chloride lead to iconazole. 5. Alkylation with orthoparadichlorobenzyl chloride give miconazole. The next one drug is tolnaftade. Tolnaftade is a synthetic thiocarbamate used as an antifungal agent that may be sold without medical prescription. In most jurisdictions, it is supplied as a cream, powder, spray and liquid aerosol. Tolnaftade is used to treat fungal conditions such as jocket, athlete's foot and ringworm. This is the structure and chemical name of the drug: 2-nephthyl-methyl 3-methylphenyl thiocarbamate. Although the exact mechanism of action is not entirely known, it is believed to inhibit squalene epoxidase, an important enzyme in the biosynthetic pathway of ergosterol. In a similar way to allylamine, telnaftate has been found to be generally slightly less effective than azole when used to treat tinea pedis, athlete foot. It is, however, useful when dealing with ringworm, specifically when passed from pets to human. Now module 5, conclusion. An antifungal medication is a pharmaceutical fungicide used to treat and prevent mycosis such as athlete foot, ringworm, candidiasis, serious systemic infections such as cryptococcal meningitis and other. Such drugs are usually obtained by a doctor's prescription, but a few are available OTC, over the counter. Antifungal work by exploiting difference between mammalian and fungal cell to kill the fungal organism with fewer adverse effects to the host. Unlike bacteria, both fungi and humans are eukaryotes. Thus, fungal and human cells are similar at the biological level. This makes it more difficult to discover drugs that target fungi without affecting human cells. As a consequence, many antifungal drugs cause side effects. Some of these side effects can be life-threatening if the drugs are not used properly. Apart from side effects like liver damage or affecting estrogen level, many antifungal medicines can cause allergic reactions in people. For example, the azole group of drug is known to have caused anaphylaxis. There are also many drug interactions. Patient must read in detail the enclosed data sheet of the medicines. For example, the azole antifungals such as ketoconazole or itraconazole can be both substrates and inhibitors of para-glycoprotein, inhibitors of P-glycoprotein, which excrete toxin and drug into the intestine. So dear student, this is Dr. Varsha Kashav takes leave from you with the hope that you have enjoyed and understood today's lecture. We will continue in our next lecture with another topic. If you want to learn more and enhance your knowledge, you may log on our website www.cec.nic.in. Till then, keep studying. Goodbye.